My areas of, let's call it like expertise or of interest are within hormone receptor positive disease and trying to discriminate the different groups that there are from a prognostic point of view and from a predictive point of view. Also within HER2 positive, we're trying to identify novel biomarkers that can help us make better treatment decisions, especially regarding anti-HER2 therapies. And also within triple negative, we're doing some studies where we're trying to dissect this group of tumors into different groups and try to find additional biomarkers beyond the ones we have, which are basically none. So on one hand, within HER2 positive disease, for example, we have identified at least 4 different subtypes within this group. The luminal A, the luminal B, the basal-like, and the HER2-enriched. We've shown in the past that within HER2 positive disease, if you are HER2-enriched by gene expression, you respond more to anti-HER2 therapy in combination with endocrine therapy, for example, or without, but even without chemotherapy. And we're further trying to pursue this as a potential biomarker that could help us identify a group of patients with HER2 positive disease that in the future they could be cured with just biological therapies without the need of chemotherapy, which I think this could be definitely very relevant. Within the hormone receptor positive group, we're trying to dissect that group, as I said before, into, again, the different subtypes and try to see if one of them responds more or less to novel agents like CDK4 and 6 inhibition. We have already data, I think, out there suggesting that within this group, the hormone receptor positive, if you're non-luminal by gene expression, probably you don't benefit from this drug, so novel strategies are needed, and we're trying to find which ones in the lab. And within triple negative, we are mostly focusing on a group of tumors that is a minority, but is a group of tumors that are probably driven by androgen receptor, among other alterations, and is our focus to try to find new targets within that group and new drugs that could help improve outcomes in a group of patients that today we only have chemotherapy. Definitely a lot is changing. The complexity is high, but I think CDK4 and 6 inhibitions are here to stay, not only in hormone receptor positive disease, but also in the other groups. Now the big question is going to be how are we going to combine these treatments? How are we going to do the sequential treatment upon progression? That's where I think the challenge is. How are we going to study these tumors as they evolve? Certainly we have technologies like ways of measuring in the blood the DNA and mutations and tumor burden, but probably that's not enough compared to lung cancer, when we have particular DNA alterations that are oncogenes. In breast cancer, probably we need to go to the tumor and study more RNA, protein, epigenetics. So that's a big challenge. So definitely what I envision is that we need studies that upon progression, CDK and 4/6 inhibition, for example, we can study that tumor and start dividing the different groups and running trials in these different settings. I do envision a setting where really things are changing. From a DNA perspective, I do believe estrogen receptor mutations are also probably going to make an impact in the clinic, as well as HER2 mutations. So now the question is, once we have these drugs, how are we going to do these combinations? Depending of the biology of the tumor, should we start with one, with the other one? So the sequential is going to be critical. So a complex scenario, but at the same time an exciting scenario. Well, I think one of the main take-home messages is that if we want to continue evolving and improving the outcome of our patients with either early breast cancer or metastatic disease, it's critical that we do a lot of translational studies. Of course, these are not always readily available, but unless we do that, it's going to be very difficult to really move the field forward. So participating in clinical trials, in translational projects today, and now especially that we're identifying subgroups of patients that are a minority, either we do this in collaboration, not only in Europe but even across the Atlantic, we won't be able to answer important clinical questions. So I think that's my main take-home message.